Nuts and seeds have always been celebrated for their healthy fats and nutrient density, but aging science now reveals an even more encouraging story. Their unique combination of vitamin E, selenium, polyphenol, and plant-based antioxidants plays a meaningful role in protecting telomeres, the tiny caps at the ends of chromosomes that shorten as we age. Telomeres function like a biological clock, and their rate of shortening reflects the cumulative stress our cells endure over time. What makes nuts and seeds so promising is that their nutrients interact directly with the oxidative and inflammatory processes that drive telomere erosion, helping preserve the structural integrity of DNA and support a more youthful biological profile. The evidence, while still emerging, is consistently positive. Regular consumption of nuts and seeds corresponds with healthier telomere markers, better metabolic balance, and slower systemic aging. Vitamin E is one of the standout components in many nuts, particularly almonds, hazelnuts, and sunflower seeds. This fat-soluble antioxidant integrates into cell membranes, making it especially effective at neutralizing lipid peroxidation, a major source of damage to telomeric DNA. Because telomeres are exquisitely sensitive to oxidative stress, vitamin E's membrane protective actions help keep them stable and reduce the rate at which they shorten during cellular replication. Studies have shown higher vitamin E intake correlates with longer telomere length in adults, while deficiency is associated with accelerated shortening. This protective effect is amplified by the fact that vitamin E supports stronger cellular repair systems, allowing DNA repair enzymes to work more efficiently and prevent the accumulation of lesions that compromise chromosomal structure. Selenium adds another powerful dimension to telomere support, especially abundant in Brazil nuts. Selenium is essential for the production of glutathione peroxidase and other selenoproteins that control oxidative stress at a deep enzymatic level. These enzymes prevent reactive oxygen species from attacking DNA, including the vulnerable guanine-rich sequence of telomeres. Individuals with higher selenium status tend to show lower oxidative damage markers and more stable telomere lengths. In some studies, even modest increases in selenium intake, such as 1 or 2 Brazil nuts per day, improved antioxidant enzyme levels and reduced DNA oxidative stress, creating conditions favorable to telomere preservation. This gentle yet powerful influence on cellular redox balance contributes to a biological environment associated with slower aging. Polyphenol and other phytochemicals in nuts and seeds add another layer of protection. Walnuts, pistachios, pecans, flax seeds, chia seeds, and sesame seeds contain diverse polyphenol that activate antioxidant pathways, improve mitochondrial efficiency, and calm chronic inflammation, three processes directly linked to telomere health. Chronic low-grade inflammation accelerates telomere shortening through both direct oxidative damage and increased cellular turnover. Polyphenol help interrupt these processes by modulating NF-kappa B and activating NRF2, thereby reducing inflammatory gene expression and boosting the cell's internal antioxidant capacity. Over time, this shift in molecular tone supports a more stable telomeric environment and helps preserve DNA integrity. Research connecting nut and seed intake with telomere length is highly encouraging. Several population studies consistently show that individuals who regularly consume nuts tend to have longer telomeres, lower inflammatory markers, healthier lipid profiles, and stronger metabolic resilience, all traits associated with slower biological aging. Walnuts, in particular, have been highlighted in longitudinal research for supporting telomere length and cognitive function in aging adults. Similarly, mixed nut interventions have demonstrated improvements in oxidative stress biomarkers, reductions in C-reactive protein, CRP, and enhanced endothelial function, all of which indirectly support the cellular conditions needed for telomere maintenance. These benefits are not limited to antioxidants. Nuts and seeds provide plant-based omega-3s, magnesium, L-arginine, and fiber, each contributing to a metabolic landscape favorable to long-term cellular health. Omega-3s help reduce inflammation and improve mitochondrial function, Magnesium supports DNA repair and prevents genomic instability, and L-arginine aids blood flow and endothelial health. Together, these nutrients create a systemic environment that shields telomeres from stress and enhances the body's ability to maintain youthful cellular operations. Perhaps the most encouraging aspect is how practical and enjoyable these foods are. Adding a small handful of mixed nuts to daily meals or incorporating seeds into yogurt, salads, smoothies, or baked dishes provides a steady supply of telomere-supportive compounds. Unlike supplements, whole nuts and seeds deliver these nutrients in a balanced matrix alongside healthy fats and plant compounds that enhance absorption and create synergistic effects. The accessible nature of these foods makes long-term adherence realistic and sustainable. Nuts and seeds won't freeze telomere shortening entirely. Aging is a natural process, but they help slow the pace by reinforcing the fundamental defenses that keep DNA stable and resilient. Through the combined power of vitamin E, selenium, polyphenol, and supportive micronutrients, they offer a deeply positive and scientifically grounded approach to protecting telomeres and promoting a more youthful biological profile. As part of a balanced diet, they become small but powerful allies in sustaining vitality, cellular integrity, and graceful aging over the long term.